Folks, welcome to the afternoon sessions. This is the open session 8 monitoring trajectory. My name is Nick Dickinson, just keeping the time this afternoon, and we're running about a minute late, so hopefully we'll pick up, but I will allow you to run in as speakers. The first talk this afternoon, a series of interesting presentations. The first one is from Sophie Cross, Curtin University, on responses of fauna to mine site restoration.
So my PhD is looking at how animals respond to mine site restoration, in particular how varanids or monitor lizards respond behaviourally. This talk is just covering one data chapter, which is broadly looking at how animals respond as a whole, the whole community. So mining has a huge environmental impact. As you can see here on the right, it can alter landscapes from relatively intact and undisturbed systems into fundamentally altered and inhospitable landscapes on the left. So this has a huge effect on animals, as it can directly impact them in terms of dispersal and gene flow, but also has a number of indirect and enigmatic impacts, such as noise and vibrations. So across Australia, there have been just over, somewhere around 60,000 mine sites identified as discontinued, yet although restoration is a legislative requirement, the number of these known to be restored and officially closed is quite small. In assessments of mine site restoration success, monitoring plants is quite common, but monitoring animals is far less common, with the assumption that they will return to these habitats following the return of vegetation. This is commonly referred to as the field of dreams hypothesis, as in practice actually recovering animal communities to pre-disturbance abundances is exceptionally difficult to achieve. So although monitoring animals is less common, among studies that do assess animal responses to mine site restoration, there's a heavy bias towards point sampling of particular taxa like ants and birds, which are generally quite easy to sample. However, when it comes to the whole animal community, little is actually known about how animal communities as a whole respond to mine site restoration. And just sampling particular taxa at particular points in time may not actually adequately reflect how that habitat is being used. So, for example, are animals actually using restoration areas long term, and are they supporting breeding populations, or are these areas only being used opportunistically and just, infrequently visited? So, especially in terms of high-order predators, little is known about how they respond to mine site restoration, very likely as it requires a lot of time and a lot of money to actually assess these animals. But high-order predators generally rely on large home ranges and also large landscape scales. And so if we can understand how they respond to mine site restoration, this may provide an indication of how animal communities respond over far larger landscape scales. So my study, this first chapter, is basically looking at how animal communities as a whole respond to mine site restoration and how the enigmatic impacts of mining actually impact these communities. So this is particularly looking at in terms of the distance of the sites to active mining, as well as differences between reference and restoration sites. So my study sites are located in the Midwest region of Western Australia at Carrara mine site, which is about 450 kilometres northeast of Perth and a couple of hours east of Geraldton. These comprise one reference-only site, which is located 3 kilometres from the active mine pit, and 2 restoration sites with adjacent reference vegetation, which are located 8 and 14 kilometres from the active mine pit. So this study comprises 2 main components. One of them is my main field season, which runs from September to November, and this is when reptile activity is beginning to peak. So to date, we've completed 1 field season, which was last year, and we have another currently underway at the moment. So the main field season basically involves intensively camera trapping each site, 1 site per month, And to do this, we set remote sensing cameras in both reference and restoration areas, and they basically trigger whenever an animal passes within the field of view, and this is both during daytime and night. So to date, this study has had a total trapping effort of just over 2,000 days, and that's basically calculated by the number of cameras set by the number of days set for times by the number of sites. The second component is a more seasonal and long-term study, which was basically designed to see not only how animals are responding to mine site restoration between each site, but also how they respond over longer timeframes and between seasons. So this basically ran for 7 months between February and August this year, and it involved setting cameras at fixed locations at our 3-kilometre and our 14-kilometre sites. That particular study had a trapping effort of just over 5,500 days. So just to put it in perspective for the length of time that cameras have been at our site, the combined trapping effort for both the main field season and the seasonal cameras to date has been just under 8,000 days, or just under 190,000 hours. So this is kind of preliminary results, as the study is still ongoing, but to date we've had quite interesting trends in the data that we've been picking up. Most notably is that animal detections significantly increase as you move further away from the active mine pit. And this was true for both 2017 and 2018. Interestingly, in 2017, the actual species diversity between sites didn't significantly differ, but there were significant differences in 2018, with double the amount of species detected in our furthest site from mining compared to the site closest to the active mining. So this may suggest that not only are enigmatic impacts of mining, like the noise and vibrations, likely impacting animal communities, but also suggests the need for far longer term studies to actually see what is going on in the animal community. This slide basically breaks down the previous slide into detections by vertebrate class, and as you can see here, the proximity to mining pretty much affects the whole animal community. We found particularly birds and mammals tended to be more likely to be detected further from mining, and this was true also for reptiles, however there were low detections. So as with proximity to mining, we found interesting trends between reference and restoration sites. Again, we didn't find significant differences in the actual total species diversity, but there were significant differences in the actual vertebrate class that was being detected. So we found that reference sites were far more likely to have birds detected in these areas, whereas restoration areas were far more likely to have mammals. However, a vast proportion of the mammals that we did detect in restoration areas were kangaroos, and they were present in higher numbers than they were in reference sites, which may suggest that these areas are over-facilitating the return of kangaroo populations. And this is very likely due to the increase in the availability of seedlings that they can graze on. Interestingly, in both reference and restoration sites, we found quite low detections of reptile species. However, again, there seems to be quite an important impact of the enigmatic effects of mining, in that we detected them almost exclusively at our site furthest from active mining. So lastly, we also found that native and feral species tend to be affected by mining and mining activities to different extents. Feral species didn't seem particularly perturbed by anything. They were present no matter what the state was, whether it was reference or restoration vegetation, and they didn't really seem to respond too much to the distance from mining either. But native species appear to be far more selective of their habitats. We detected the critically endangered malleefowl, which is this bird on the bottom right, and as well as several species of monitor lizards, and these were detected almost exclusively at our site furthest from active mining activities. One fantastic result was the detection of a couple of parentes, which are Varanus giganteus, the bottom left, which is a species of monitor lizard. And we detected a few of these in the restoration area of the site furthest from active mining, which may suggest that these areas are beginning to facilitate native populations and may potentially be bringing back resources to support their growth. So just to summarise, there was a huge impact of the distance from mining on detection likelihood of animals, and we found that as you get further away from the active mine pit, you're far more likely to be detecting animals and very likely at increased species. And this may be partly due to noise and vibrations associated a bit with being close to the mine pit, which could potentially impact things like monitor lizards to a huge extent, because a lot of these species, especially the ones that we have on our site, they tend to burrow, and if you're burrowing in an area which is constantly vibrating, it's probably not really conducive to living there long term. Also, while restoration does appear to be facilitating the return of some animals, it doesn't seem to be bringing back whole communities just yet. And so this may indicate that to actually recover your species diversity, you need to invest significantly more time and resources and monitoring into these particular areas. So in conclusion, there is a huge need to increase the monitoring of animals on mine sites to assess their restoration. And this not only needs to incorporate your standard bioindicators, like the things like the ants and birds, but it has to include your wider animal community. And this needs to be done over far longer time frames, especially over different seasons where you might be picking up differences in communities, and also at far larger landscape scales, because there are obviously effects which impact animals to varying degrees as you move further away from the mine pit. And lastly, including the effects, the enigmatic effects of mining, like your distance from the mine, the noise, the vibrations, the distances from roads, is essential in monitoring restoration, as it may also include, you may be able to develop far more targeted and effective monitoring plans if you incorporate these further effects. And just a couple of acknowledgements to Centre for Mine Site Restoration and my supervisors, and especially to Carrara Mine Site and the environmental team for letting me stay on site for most of the last few months. And also a couple of field assistants and the ESA and Hosworth Wildlife Research Endowment.
Thank you, Sophie, and thank you for keeping to time. We do have time for a few short questions.
The effects of vegetation cover, in proximal to the mine, usually there's a lot of stripping that occurs around there. Has there any correlations between that?
We're going to be starting to look into that with a later study, which I'm planning to do in November, basically assessing the extent to which the metabolic and predation risks might be impacting an animal's choice between a more open or a more covered landscape.
Yeah, the predators, that's what I was thinking, wanting more cover.
Yeah, so that's definitely going to be something that we're looking at this field season.
There's a question at the front.
Thanks. That was really great. So you did just under 190,000 hours of tracking, is that right?
Yes.
Is that right? Okay. How long did it take to go through all of this?
Oh, then to actually go through the data, a long time, especially when you get a camera which decides that branches moving is a fantastic thing to take photos of. I literally had one camera which took 25,000 photos of branches moving and had maybe like one kangaroo in that. But the problem is that you have to still go through all of those because you may potentially miss something. Thankfully, I was able to do it so that I was up for a day or two each month and collected it, and so it wasn't so onerous at the end. But it still took a lot of time to sit through all the images.
You said that current approaches sort of
figures, can.
things like flying intelligence rely quite
See if you can see it.
having hollowed out blocks that they can live in and hide in the crevices. So I think that actually helping to facilitate certain groups may through maybe artificial hollows or housing.
Oh, this is the old one,
would be
isn't
very
it?
critical because they're not really going to come back to an area if they don't perceive there's any safe place that they can live.
I do that briefly today. Does that have any impact on the mining company that's down there? Did they know about that before?
Yeah, so our site particularly does extensive monitoring for power as well as lessons mining testing. Basically, any recordings that I find have to go back to the environmental team because they report all sites. But they have quite a lot of data on existing mounds to the mound which they go back and monitor every year to see which ones are active or which ones are not still being used. So they definitely do have to incorporate a lot of those. It's not so much a surprise, but I don't think they have a lot of breeding pairs around site. So one of the pairs that we look at in Canada was like quite young, which they seem pretty pleased about.
Okay, we have to finish there. Thank you, Sophie. Sorry for the slight delay there, but subsequent speakers, if you could come out quite quickly, some of the files seem to be missing this afternoon. We'll check will be there as soon as Brendan finishes speaking. Luckily, Brendan has his spare one on a USB station. Brendan is from consultancy National Bushland Ecology, on the Sunshine Coast. To you.
Yeah, good afternoon, guys. Thanks for joining us. I'm going to talk today on defining the parameters of successful bush regeneration outcomes. I live up at Malula Valley on the Sunshine Coast. I've been doing bush regen in the local area since about '94, and yeah, I'm pretty passionate about it. And I just want to have a chat today about what the exact parameters of success are, because there's a long range, there's a lot of approaches that people use, but I think it's really important to clearly define exactly what we're trying to achieve. So bush regen is comprised of sort of three streams, really. We've got the practitioners that do the work on the ground, we've got the managers that are managing the sites, and then we've got the researchers who are actually accessing the information, the theoretical stuff behind the ecology of what we do, and also, I suppose, quantifying and qualifying the results of what we do. Personally, for me, I run my own natural bushland ecology, bush regeneration business, servicing a whole range of clients, usually local government, local landholders, national parks. I'm also a manager. I'm a Community Nature Conservation Partnerships Officer with the Sunshine Coast Council, doing bush regen works on their conservation estate with community groups. And I'm also a researcher doing my Masters here at UQ. So just over time, I think I'm in a great position to provide a bit of feedback on the industry, as I've got my sort of hat in a couple of different streams. I suppose the fundamental of bush regen is vegetation dynamics. That's what we're trying to do. We're basically trying to restore the ecology and the functionality of our ecosystems. To me, understanding veg dynamics is the most important fundamental in bush regen. The parameters of successful bush regens, what I'd like to do first, instead of actually defining what that is, so I'll give you a hint, it's one word, 11 letters. We can discuss that at the end, but what I thought we'd do first is have a chat about what are not the parameters of successful bush regeneration. Because like I said, there's a whole different range of approaches of what we do, and I think it's important to clearly define what we're trying to achieve, and these are quantifiable outcomes. So first of all, it's not the square meterage of weeds that we clear. It's certainly not the cubic meterage of weeds that we clear. It's not time on site. It's not the labour days that we have on site. It's not the number of native tubes that we plant in the ground. It's not expending our budgets. And it's certainly not a herbicide record. Even though sometimes those variables are what managers will define as an outcome. They're on the way there, but they're certainly not the parameters of successful bush regen. In the last, say, 5 years, I've really noticed that the standards of bush regen have substantially decreased in my industry. The role of vegetation dynamics is rarely discussed anymore. There is excessive use of foliar spray applications across all sites. There's a reliance on foliar spray applications to generate ecological change on site. And this is having an impact on native vegetation, which is the basis of what we're trying to restore. Through that observation of the way that those standards have dropped, I've seen that there's a few, I suppose, topics that I'd like to discuss. One of them is the lack of the creation of regeneration niches and overuse of foliar spray applications and the assessment and accountability of our works. Now, I'd like to talk about the lack of creation of regeneration niches. Like I said, veg dynamics is an essential component of what we do, and basically it's a change of species composition and structural complexity on site over time and space. When we're looking at our sites, I think it's important to try to assess what the disturbance history is of that site. We need to have these conversations trying to work out where this site is along the successional spectrum, what is the disturbance history there, what has happened in the past, because you can interpret past events, past ecological events, by looking at a site, looking at the structural complexity and the species composition on that site. You can interpret what's actually happened on site in the past. And we need to know how that site is responding to that disturbance. With the species that we're looking at, it will tell us how old that regrowth patch is. With the structural complexity that's in there, we can determine what level it is in response to that disturbance. What successional stage is it actually at? And when we're looking at that specific patch, vegetation is always trying to expand, and there's a certain suite of species that occupy those areas. Just like any sort of vegetation, there's a range of change over time and space, and there's specific species that are adapted to those external conditions and that hostile environment on the edge that wants to expand out through. So I think it's important to understand what the regeneration niches of those species are. And we need to create what those niches are on site. Through our bush regen works, we need to focus on creating those regeneration niches, those ecological and physical gradients of what those niches are. It's not just about going in and controlling the weeds and expecting the site to respond in a certain way. We need to have a look at where the reproductive material of those regeneration species are within the local landscape. Are they actually existing on site within the seed bank? Are they actually being produced in the surrounding landscape? It's all about timing also, because lots of our systems here on the sunny coast, the phenological systems that are in place, some species will only put a good fruit on every 5 to 7 years. And sometimes that changes actually through the seasons as well. So when we're actually creating these regen niches on site, we need to keep in mind what is actually in the surrounding landscape and what are the fruiting patterns of those species, and are they within dispersal distance on site? Is that reproductive material actually going to get on site? So if we create the regen niches at a specific time of year, is that reproductive material actually within the landscape, and is it within dispersal distance so that it can actually get onto site, germinate and persist on site? And we need to make sure those regen niches are created through our bush regen works. Another thing I'd like to discuss is the overuse of foliar spray applications. Now, I just want to state that I'm not an anti-sprayer. I do have a knapsack. I do use it. I've been spraying herbicide since 1994, but yeah, I want to have a comment on this only because there seems to be, it's always been part of our toolkit, but there seems to be quite an extension and quite an increase in the last 5 years of herbicide applications within our bushland areas. The history of herbicide and bush regen is because through land care, I did a lot of work in the 90s with land care, but basically we started doing re-veg on grazing properties, and so working out sort of how to maintain and look after the plants that we were putting in. Once the plants were in and the weeds came up, we sort of looked to the farmers. I remember having conversations with them and asking them, mate, how do you control the weeds in your pastures? So bush regen and that land care ethic was heavily influenced through that agricultural focus with weed control. And there's nothing wrong with that, but there's different management objectives when it comes to bush regen and an agricultural enterprise. So that's why herbicide actually has a history and, I suppose, a presence in the bush regen industry. It's an effective component of our toolkit. When I'm preparing a site for a planting, if it's a pasture, sometimes there's certain species that will only respond, especially ground cover, ground stratum species that are very abundant in areas where there's a lack of native vegetative cover. Herbicide is a very effective tool, but it's not the first tool, and it shouldn't be the first response. We should try a whole range of approaches, and then if nothing else is actually generating change on site, that's when we throw the knapsack on. My issue with herbicide is just, I've seen it just be ineffective. I've seen large areas that continue to be sprayed, the target weed population doesn't actually die, parts of it do, there's no disturbance, there's no actual creation of those regen niches, there's no discussion internally or within the team or with managers about creating those regen niches on site, and the excessive foliar spray is impacting on our sites because it's not creating the regen niches and it's actually impacting on existing native vegetation. In my role as a Community Nature Conservation Partnerships Officer with the Sunshine Coast Council, I manage about 12 reserves. I work with community groups as contractors, and a lot of these contractors are actually friends of mine. And I have discussions with them. I have to pull them up and have site meetings with them because through their bush regen works and through their restoration of the site, they're actually impacting on the existing species that are there. I've got a whole range of examples, but one very recent was probably about 3 square metres of camphor laurel seedlings down at Nambour, and contractor come through and just sprayed out the camphor seedlings. As he did that, he hit 4 Goya semi-glauca saplings that were dead. So the target weed population wasn't even effectively controlled. The camphors were there, and the existing native vegetation that was on site has died as well. So it's impacting on the existing native vegetation, and it's impacting on the future recruitment on site as well. And then when I was there for the working bee on the weekend, myself and another volley spent about 10 minutes hand weeding those camphor seedlings out. Effective target weed species mortality, creation of regen niches, and no impact on native species. It's quite simple. I'll just jump through. There's a lack of assessment and accountability, so even though we're throwing lots of money at bush regen projects, there isn't really much assessment of what's actually happening on site at all. Sometimes these are the existing variables. Like I said before, they're not parameters of a successful bush regen project, but sometimes these are what managers will talk to practitioners about. If these things are done, then the site must be good, the project must be a success. They're on the way, but they are not parameters of a successful bush regen practice. And lots of that assessment process isn't actually delivered by existing practitioners. Lots of the managers who are actually doing this were bush regenerators a long time ago. Things have changed, or they haven't actually been involved in the industry. So it makes sense to get practitioners to be regulating what's going on. Now, getting back to the parameters of successful bush regen outcomes, like I said, one word, 11 letters. There are quantifiable variables that we can talk about. I will go through these quite quickly. Effective mortality of target weed species, 100% reduction in target weed species, evident increase in the diversity and the abundance of native species over time and space, an increase in structural complexity, the movement of veg dynamics into later successional stages, and the occupational dominance of native species in all niches and all strata. Now, they're actually quantifiable variables that you can actually go out on site with a pen and paper, doesn't matter the scale of the site, and you can actually mark these things. You can do these assessments at the start of the project, 6 months into the project, five years down the project. They're actually accessible, quantifiable variables. Bush regen, parameters of successful bush regen outcomes is one word. There's a hint. It's recruitment. That's what it comes down to. If we want to make sure our projects are successful, we need to have recruitment on site. And like I said, that is an impact that's happening in the industry, is that the weed species aren't being effectively controlled, and the recruitment that manages to pop up on site is significantly and repetitively getting sprayed out through excessive foliar spray applications. And our recruitment needs to change in abundance and diversity over time, and these are just a few. This is what it's all about. Bush regenerators, quite simple creatures. We get excited about seedlings coming up on site and watching them change over time, watching them go through into different age classes, watching the diversity come through. That's a Du Boisier Myoporoides. The dispersal distance for that is quite long, and that was on the site, McCarthy Shoot Road at Mullaney, and there wasn't a single mature individual within a couple of Ks, and all of a sudden he's up on site because they have seed bank resilience. But that is me done. I'm sorry for rushing through it, but thank you very much.
Thank you, Brendan. We have time for a quick question. It occurs to me that you were really saying you need to stand back and have a think before you go in all guns blazing.
Yeah, exactly. Have that sort of discussion about vegetation. Put the ecology back in the ecological restoration.
So the SERA standard suggests using a referencing system and setting up reference plots. So have you seen that done much in the book review?
Theoretically, I mean, that's what we're doing, you know. Like when I'm talking before about regeneration niches, we know what species are likely to come up in response to what we're doing.
I think they're doing this wrong.
What system it is,
Do you use this? I do.
what the type of vegetation community is, and what those inherent dispersal disturbance mechanisms are to change the species over time. So we can relate it back to different levels of successional change within a focal or a retarded type of vegetation, for sure. Yeah.
Thank you again, Brendan. Again, to those speakers, when they wrote it up, I think they took the pen away with them, so please come over with your USB stick. If you only got one, rush out and copy, please. Next up, I'd like to introduce Sacha Jellinek, who's going to talk to us about monitoring plant survival in revegetated agricultural landscapes.
Cool. Thanks very much. Today I'm going to talk about a large-scale revegetation project which was done in South Australia, and this is part of it, which was monitoring how well the plants that were revegetated survived in that landscape. So, when you're looking at revegetation and when you're assessing how effective revegetation is, there's a number of different factors which feed into how well your plants do and how well the area survives and changes and how functional it is. And to start off in a revegetation sense, that depends very much on the site, what preparation is undertaken, the planting method, the planting context, the planting type, and also the configuration and size of the planting area. There are various temporal and spatial considerations, including climate change, and that all feeds into how flora and fauna respond in these areas, the time since restoration, and ultimately how that area develops structurally and functionally, and what the biodiversity outcomes are, and what the goals of the revegetation project are. Today I'm just going to be talking on plant survival. So this project was undertaken, as said, in South Australia. It was undertaken during the Millennium Drought, and the Coorong and Lower Lakes area, which is at the end of the Murray, is a huge system and it's a really important Ramsar wetland. And this area, when the Millennium Drought was on, the water receded in the lakes 200 to 300 metres. So it was a really huge impact, and the federal government funded a project to essentially try and restore those lakes, reduce the threats of things like acid sulphate soils, and the original plan was to do a lot of seeding within the lakes themselves. Of course, as soon as the money came through, the water came back, so the whole project had to be changed. So this is a talk on that change, which was a large revegetation project around the perimeter of the lakes and the Coorong. So it was a huge site, 142,000 hectares, and over 4 million plants were put into this landscape. 4 million terrestrial plants. There was also aquatic restoration going on as well. So the areas that were revegetated, and that was revegetated by both community groups and the government and through consultants. And more than 160 sites, 1,300 hectares were revegetated, and they were all revegetated with tube stock on public and private land. And tube stock were used mainly to allow the community to be more involved in the revegetation. As I said, over 4 million plants were planted and over 200 different species were planted across this landscape. And they included trees, shrubs, grasses, herbs and sedges. And so the monitoring was undertaken at each site for each year, and we looked at survival directly after planting, so 2 to 3 months after planting, which is spring, and also in the autumn, so 8 to 9 months after planting, after the first summer, to see how well plants survived. And as I said, we surveyed different sites in each year because that's how the planting was done. It was planting different sites in different years. So to survey these areas, we went out and we put a whole lot of random transects across the revegetated areas, and we used 50-metre transects. We went and recorded all the live and dead plants and identified them if we could to species level. And we used, so this is what the monitoring looked like, 50-metre transect, looking either side, 1 metre either side, and identifying all the plants in that transect. And we did a lot of transects, over 4,000 I think. And these are some of the results. So this is using a generalised linear mix model, and looking at the differences between autumn and spring, we can see that spring so this is directly after planting, you do get a decline in plant survival, so this is probably most related to planting shock. Yeah, about 13% decline in the species that were planted. After the first summer, there's a further reduction of about 18%. So overall, you have between 65 and 70% surviving after the first summer in tube stock revegetation. When we looked across different years, we saw that although 2013, 2014 did have slightly higher survival, generally it was surprisingly similar across the different years. Although you still had those seasonal differences between spring and autumn. And we found that the revegetation was substantially impacted by temperature, so the mean temperature across the site, but it wasn't that influenced by either rainfall or the maximum mean temperature, which was interesting. And we also found that where the region where the revegetation was undertaken in was really important. So we defined region looking at soil mapping to determine, broadly speaking, how the different areas differed from one another. So we had the Mount Lofty Ranges area, which is the purple, the Lower Lakes area, which is the more reddy colour, and the Coorong, or the southeast, which is the green colour. And you can see we had a lot more survival around the Lower Lakes, probably because that area is in some ways more fertile and lower. Along the Coorong, we got a lot lower survival, probably because that landscape's a lot sandier. Up in the hills, along the Mount Lofty Ranges area, we had moderate survival. And although soil didn't play a substantial role in the survival of plants in this project, we did find that, especially in deep sandy sites, survival did decline. And when we looked at how well different trees and shrubs and grasses survived, we can see that trees and shrubs and also herbs survived relatively well during the plantings, had between 80 and 90% survival, whereas grasses had a lot lower survival, as did sedges. And although there are 200 species planted, so it's rather difficult to show a graph of how each one survived, these are some of the most common species used in that revegetation project. So you can see some of the poas didn't survive so well, whereas some of the eucalypts and encolenas, they survived really well. And I was interested to see, okay, we put all these plants in the landscape in varying densities. Overall, is that the communities that we're trying to create after the first summer, are they still there? So I ran a cluster analysis just to see what would come out of that. And it was really interesting to see, broadly speaking, we had a lot of, in the initial planting, there was a lot of different species and a really broad community that was planted. And after spring and after the first summer, that really condensed. So what you're actually putting out into the environment isn't actually what's surviving necessarily. So what are some of the factors that might influence this survival? I've talked about planting shock, so initially once the plant's going to the ground, extreme temperature and rainfall events, although we didn't seem to, maximum temperature, maximum mean temperature didn't seem to be a major driver in this case. The age and the condition of the nursery stock was one thing we noticed was important to monitor. So when the plants were first planted, to see if the plants were planted well and that the nursery stock was good. If the plants were guarded or not. Generally the plants were guarded, but in some cases they were planted with different types of guards, and that also influenced how well these species survived. The soil moisture in the area, both prior to and during planting, and how much weed competition there was or grazing from stock which got into the site or from other herbivores. And there are other things like mechanical damage from winds and sand, non-wetting soils on sandy sites, and also something which usually isn't looked into a great deal, what sort of previous land use history, how does that impact the site that you're revegetating? So in conclusion, after the first 2 months we saw a decline of about 13% in the plant survival, and then a further 18% decline after the first summer. So overall we had a survival rate of about 70%. There are multiple factors that influence this, but some of the main ones that the analysis showed was temperature and the region that these were planted in. And we found that there was areas that we're trying to restore didn't actually resemble that what was planted in the first place. And a point that's been made over and over again in this conference is that monitoring is essential if we want to adapt and learn from the restoration projects that we're undertaking. And one of the things is we need a standardised survey method so we can actually compare between different sites in different revegetation programs. And ideally an overarching database where all this information is stored so we can actually adapt and learn from revegetation projects across the country. Thanks.
Thank you. Standardised sampling methodology there. Yes.
I just wanted to ask you, from a planting perspective, there's usually a continuum from, say, a framework to a maximum diversity in terms of the number of species that you into a site.
Yeah.
You had a lot of species there. Are there any learnings you've taken away about whether a framework planting of much less species to sort of capture the site might have given you greater survival in the long term to then do supplementary work later on?
That probably would have boosted survival initially, but you may have, I mean, the likelihood of going back to these sites after they're planted, especially in this project, which was, I mean, it was five years long, which is quite rare in revegetation projects. The ability to go back to sites is fairly limited usually. So I get what you're saying about trying to get some which are going to survive over the long term, and then supplementary planting.
So yeah, just got to turn 2 slides off.
And I think that's
before we start it up.
Maybe a small land care context, but this is a really big, broad project, and there wasn't the capacity for that, I guess. So it depends, horses for courses, I guess. It depends on the project.
Okay, thank you, Sacha. I think we're okay, are we?
Yeah.
The projector hasn't backed up.
I'll put it back on in a second. They're off already. Sorry, cool.
We're
Yep.
not too stressed for time, apart from people are swapping rooms because we finish a little earlier than the other session.
No worries, thanks.
Here we are, back on track, and there will be room for discussion. So if we're a little over time, we can talk afterwards. But next, I'd like to introduce Tim Whiteside, who's from the Department of the Environment and Energy in Darwin. He's going to talk about the Ranger Uranium Mine. Thanks, Tim.
Okay, thank you. So I work for the supervising scientists. We've had a few presentations already yesterday. Supervising scientists' main mission, I suppose, is to protect the Alligator Rivers region. That's right, over here. Alligator Rivers region in the Northern Territory from the environmental impacts of uranium mining. So my talk's about how, as the mine is heading towards closure, how we can derive some measures that the mine can aspire to as they do their rehabilitation. Okay, so the Ranger Uranium Mine is surrounded by Kakadu National Park, which is World Heritage listed, and it is also upstream from Ramsar listed wetlands, so it's quite an important environmentally and culturally sensitive area. It's got a uranium mine in it. The Alligator Rivers region includes Kakadu National Park and in Western Arnhem Land. It's got a strongly seasonal climate in the tropics, and we get between 1.5 and 2 metres of rain per year, most of which falls between December and March. Okay, so that's Ranger Uranium Mine there. Okay, so Ranger Uranium Mine, the operations are ceasing in 2021 and rehabilitation activities are to be completed by 2026. The mine operations are subject to the environmental requirements, which are set out by the Commonwealth under Section 43 of the Atomic Energy Act, and they align with the Environmental Protection Act for the Alligator Rivers region. And the ER that I'm concentrating on today is 2.1, which says the company must rehabilitate the Ranger project area to establish an environment similar to the adjacent areas of Kakadu National Park. So, what is similar to adjacent areas, which is the main focus of this presentation? The adjacent areas are mostly eucalypt-dominant savanna woodland and open forest, which are intersected by Melaleuca species-dominated drainage lines. Savanna typically consists of discontinuous tree cover and mostly annual grass understory. Okay, so the top picture is a picture from the ground and the bottom one's a drone picture. But you can see that it's not a very homogeneous cover. Okay, the tree cover in savanna is variable over space and time, and the drivers for that change are things such as fire, grazing, herbivory, particularly by feral animals such as water buffalo, Extreme weather conditions, cyclones can knock down a bunch of trees, and that affects how many trees there are in an area, obviously, and the land use history of the area. So a lot of Kakadu prior to being a national park was used for traditional land use practices, but also for buffalo hunting and cattle grazing and uranium mining, of course. So how do we capture the variability in the savanna to inform ecological restoration and closure criteria? So looking at this question, the methods were to do a time series analysis involving historical area photography and satellite imagery, using an object-based image analysis, which extracted woody cover, a surrogate for tree cover, and calculating the proportional cover per hectare and also for a land unit polygons. The analysis of the time series included looking at change analysis, did some generalised linear modelling and photometric analysis. Okay, and then I'll talk about the implications of this for ecosystem restoration and closure criteria. Okay, so even though the area is quite remote, we do have a good historical aerial photo archive. It's nearly decadal, so it dates back to 1950. Obviously the quality of some of these aerial photo sets is not great, but we do have good coverage from 1950 to 2004 with aerial photos, and also do have some high resolution satellite imagery for 2010 to 2016. So we've got 66 years of data to look at around the mine. Well, the mine actually wasn't there in the first 4. Okay, and so we're still looking at the adjacent areas or the surrounds. So to restrict that a bit more, we've completed a 10-kilometre buffer around the mine. And then we looked at polygons in some land units created by Wells way back in 1979 that were upland terrain, and it was slopes less than 5%, which has a similar topography to the intended final landform for the Ranger mine that they're putting together. I must note that it does have a different substrate. Most of this are sandy loams, and the landform of the final landform of the rehabilitated mine will be waste rock. But these are the main classes of land units that we focused on. So using the historical aerial photography archive and the satellite imagery, all the photos were stitched together into mosaics, so they're all geo-registered to each other. And then at each level, a vegetation index was created. For the red, green, blue, it was typically the triangular green vegetation index. For the infrared data, it was using the normalised difference vegetation index. And for the grayscale images, we used some texture measures. Then we masked out the land units using the boundary to confine the analysis, extracted the tree canopy for that polygon, gridded up the polygon and created 1 hectare cells with tree cover in them, and then from that we could create proportional classes. Okay, so this is the results over the time series. As we scroll through, you'll notice that this area is relatively stable, this area changes a bit more variably. So sorry, on the left is the 1 hectare cells and on the right is the land unit polygons. So we can see that over time it does change quite drastically. Yep, in 2010, because there's a limited area of coverage that's clipped off there. Oh, and don't worry about that little circle, it's not supposed to be there. So if we look at the mean cover of variability through time for that time series, we can see that per 1 hectare cells, the mean cover is slightly higher than for the per polygon level. And the reason probably for that is that The per polygon one was based on number of polygons, not the area of polygons, so a lot of the small polygons probably are influencing the values there. You can see also that where there's an increase in the temporal resolution of the imagery or the data, you get more resolution in the percentage cover. So it's something that needs to probably be considered with closure criteria, is that you can't assess it based on one date, because it may change over time, of course. So we look at now the distribution of cover classes over time, and we can see that for the per hectare cells, we've got a sort of a main peak in the 30 to 40% cover class, and that's the same for the polygon one. With the polygon one though, you can see that there's a couple of peaks moved across to lower classes, and there's a bit less in the higher cover there, and that's probably because of the influence of those low polygons and averaging it out. Okay, so we can see the size of the unit of analysis does influence the cover class distribution, so the polygons are bigger and they tend to not have the extremes, I suppose, of the other one. So therefore, to be similar to surrounds, a restoration plan would need to, or closure criteria need to consider a distribution of cover. There's no one figure. Okay, so the photometric analysis was also undertaken to look at what happens to objects over time. So if you've got an object that's a tree in one time frame, does it still exist in the next time frame? Has it grown? Has it shrunk? So there's three main metrics that were assessed, which are growth, stability and decline. We can see here that not many objects actually stayed stable. So there was a lot of decline and a lot of growth of objects. So you can see that that just does show that the plate that is quite variable. And so closure criteria will need to encompass this range in cover. Okay, so conclusions from this are that cover is indeed variable over space and time, and that's consistent with previous work in the area, such as some work done by Brett Murphy and a few others previous to that, David Bowman and Carolyn Lehman. The ER, similar to surrounds, has the dynamic and spatial considerations, so there's no single value that can be used to describe woody cover for the surrounding regions. Landscape analysis at 1 hectare resolution captures greater variability than at polygon level and describes a change in detail. So it might be the preferred unit of analysis. However, at both 1 hectare and polygon levels, most of the site has tree cover of between 20 and 50% for any given date. And if anyone was at Jay's talk yesterday, he was talking about overstory from the 7 reference sites that we've done, having a cover of about 34%. Yeah, so that's pretty consistent with this. So information on woody cover, including distributions of cover class, will be used in the Ecological Restoration Rehabilitation Standard for the branch to inform mine closure. Mine closure criteria for woody cover should aim to follow a similar distribution. Okay, and this work provides measures of woody cover for all species over a large area, so there's no species discrimination. It's pretty near impossible to do with that data set. So species metrics will be provided by the work done on the reference sites described in the previous talk. Okay, so the next steps, currently there's ongoing analysis looking at the inferred drivers of change by comparing it with other data, and preliminary results show that fire is probably the biggest driver of change in the area. And also we're doing a time series analysis of riparian cover in the region, which will inform planting of wetter areas that will form on the rehabilitated landform on the toe slopes. That's it. Sorry. One more thing. We do have a suite of rehabilitation standards that have just been released on the 11th of September. The link there, and you can see that work.
Thank you, Tim. We have time for one question, if anyone would like to ask one. I was wondering how, in the future, which is what we're really thinking about, how better surveillance methods will kind of move this on.
So a couple of the guys did some talk yesterday are using drones, so we've got a suite of drones, so we'll be able to do large-ish areas, probably won't be able to cover that in the other area, so maybe every now and then maybe there is high resolution satellite imagery that will be available to look at that scale, probably.
Thank you very much. Also, there's a question here.
What's the soil depth likely to be after the rehabilitation?
There'll be no soil.
So, how much I understand the substrate's rock.
So I think it was here earlier. There's a presentation on the work that they're doing on the mine, from the mine in the next session, something like that. So what they're doing is they're growing, have shown that they can grow trees all the way from this one over there. This one, they do grow reasonably well. So but whether it'll be similar to surrounds is still.
Close this one. Close it.
Something that needs to be worked out.
Hello, that's the one.
Looks like something similar.
Is that what we got?
Thanks again, Tim. The final paper in this session is by Lachlan Charles, and he's from here at UQ here in Brisbane, and he's talking about seedling growth responses to species, neighbourhood, and landscape scale effects during tropical forest restoration. Over to you, Lachlan.
This doesn't seem to be.
That's freezing a bit.
Hello, my name is Lachlan Charles, and I'm here to talk about seedling growth responses to species, neighbourhood and landscape scale effects in a tropical forest restoration. So in response to global deforestation and land abandonment in many tropical regions around the world, we're seeing an increasing need to design effective restoration approaches. And while we actually have made kind of extensive ground in restoration techniques, we are still seeing that variability in success of restoration projects. Now this is obviously very upsetting, especially given the amount of money, time and effort spent setting up these projects. So one of the, I suppose, the main reasons why we're seeing this variability in success is many barriers that can impede seedling performance. And these can occur across different spatial scales. So for example, on a landscape scale, we're looking at things like temperature and precipitation. Moving down into more of a site scale factors, we have topographical factors such as slope and aspect, also distance to the forest, and underlying soil conditions. When we move even further down into more of like a neighbourhood scale factors, here we see that competition both between neighbouring seedlings and resident grasses can also impede seedling performance. So there's really a lot going on. So one way to kind of minimise these the effects of these barriers is to select species based on their responses to these barriers. So how do we do this, and which species do we select? So I suppose an increasingly common tool used to select species is the use of plant functional traits. So broadly, plant functional traits are just physiological traits that can influence the survival or growth or fitness of an individual within a given environment. So here is just some common traits, for example, wood density, and these are the function that they actually perform. So again, wood density, stem structure and hydraulics, leaf area is resource acquisition, seed mass establishment and germination success, and adult maximum height, growth and competition. So in essence, we're actually wanting to use these plant functional traits to really kind of predict species growth responses. So to do this, the idea about this study was to identify, firstly, which factors are influencing seedling growth in a restoration planting. And so the specific questions we asked are how important are landscape, site and planting conditions to seedling growth? Are growth rates influenced by the density and composition of the surrounding seedlings? And finally, do functional traits explain variation in seedling growth? So this study was conducted in the Thiaki Creek Nature Reserve. This is in the Atherton Tablelands in the wet tropic region of far north Queensland. And this study gives you kind of a nice example of the varied topography of the site, which is dominated by quite a few grass species. So in 2011, 28,000 rainforest seedlings were row planted in a replicated block design experiment where we had 8 blocks, and within each 8 blocks we had 8 50 by 50 metre plots. So again, just looking at within each block here, so this experimental design we had 2-level density treatment. So for example, we had a high density of seedlings planted, which is 1.7 metres from each other, and we had a low density, which was 3 metres spacing between the seedlings. This was crossed with a 3-level diversity treatment, so we had just 1 species, a monoculture in a plot, 6 species, and also a high diversity treatment of 24 species. Again, in these plots, we also had 2 control plots where no seedlings were planted. So again, this is just to give you a visual representation of what they look like on the ground. This is one of the steeper plots, and the rows here that were sprayed out, so where we could actually plant the seedlings. So for species selection, we selected a total of 24 locally endemic species nested within 6 families. And these species were selected over a variation of their successional stage. For example, we had pioneer species and later successional species, and also variation in functional trait values such as wood density, seed mass, and adult maximum height. So every 6 months for the first 3 years, we measured diameter and height growth, and also the survival status of each seedling. And this was done within a 25 by 25 metre subplot within each plot to try to avoid any kind of edge effects. So we modelled diameter and height relative growth rates as a response to multiple variables across different spatial scales using linear mixed effects models. So again, here's some of the self-explanatory. So on the landscape, we had average daily temperature, mean daily precipitation. For site variables, or site scale variables, we had slope, aspect, distance to the nearest forest, and soil nitrogen. In the local neighbourhoods, we had average basal area and average height and richness of all stems within a neighbourhood, which we classified as basically all stems within a 5.3 metre radius of a focal stem that we were looking at. Species level variables, we looked again at the functional traits listed here. And importantly, given that tree growth is known to vary with stem size, we actually included a 2-way interaction with the between the starting diameter and height size against all other variables. So this was like basically a size covariate that we needed to include. So results. So the overall results, after 3 years, we did see quite a large variation between diameter and height. So as you can see in these two pictures here, these seedlings are the same age, so there's quite a large variation. We also saw that growth was influenced by many factors across different spatial scales. And importantly, that the magnitude of these effects was really dependent on the initial seedling size. So having a look at some of these specific factors, so here we're looking at a landscape scale factor of temperature and precipitation. So before I start, you'll see here on the y-axis we have on the left here diameter growth, and on the right height growth, and on the x-axis here we have these size covariates, so the starting stem diameter and also the starting height. So basically what we're seeing here, if we look in at diameter growth, that diameter growth is actually faster during warmer periods and cooler periods. So that's kind of self-explanatory. This is what we kind of expected. And what we see with height is also that height growth is faster during wetter periods and cooler periods. Again, this is what we would expect. But what is interesting to note though, for both diameter and height growth rates, is that it really depended on the starting size of the seedling. So for example, here we're seeing that larger seedlings actually had increased growth or faster growth for both diameter and height. And what is interesting here, especially if we're looking around this part here, so if you're a larger seedling to begin with, it actually tended to reduce kind of any effect of water available, mean daily water availability. So this would kind of suggest that if these seedlings actually manage to grow quickly and establish early, there's some kind of benefit in that, ongoing benefit. So moving down to more site scale here, and here we're looking at plot slope. So again, the axes are exactly the same as the previous graph, and what we're seeing here for growth, diameter growth on the left, is that diameter growth was slightly faster for species growing on flatter slopes compared to steeper slopes. But again, we're seeing here that this really also depends, the magnitude of this effect depends on the size, the initial size of the seedling. And again, when we look at the height model, a somewhat similar pattern. However, there's not much growth, not much variation in growth on flatter slopes, but on the steeper slopes, it benefited you if you were a larger seedling, to begin with. So moving down into the neighbourhood scale, so diameter growth there wasn't really much difference between if you were in a neighbourhood with a large basal area or small basal area. But again, we see this size covariate, size dependent effect here, that larger seedlings grew better, or grew faster, sorry. We see a slightly different pattern here in the height growth in the sense that if you were in a taller neighbourhood, so if you were planted in a taller neighbourhood, you tended to do better. So this kind of positive relationship between your neighbourhood basal area and neighbourhood height kind of could suggest a few things. So first of all, I suppose the first thing we can note is that competition between the actual seedlings is probably not happening at this stage. It's still very weak. And if you remember back to some of those photos, these seedlings are still very small, so competition at this point is probably not happening. Okay. But also there might be some kind of positive density dependence effect going on here. So if you happen to be in a neighbourhood that you're all doing very well, you might be basically outshading localised grass species, thereby increasing the competition with grass and furthering your own growth. Or, which is probably more likely, we might just be seeing like these neighbourhoods are sharing a growth response to some unknown environmental variable that we didn't pick up. So finally, going down to the lowest level on the species scale, here we're looking at wood density. So again, diameter growth rate was faster for species with low wood density, which again we would expect, because especially in tropical plants, fast-growing species do tend to have low wood density. And when we look at the height growth, is again a slightly different pattern. While we are still seeing that height growth is quicker for low wood density species, we're seeing this kind of very moderate increase in height growth for high wood density species. And I suppose this can be kind of put down to the fact that many high wood density species tend to allocate resources to their stem structural support rather than shoot growth or height growth. So in summary, growth was influenced by many factors across different scales, and the response of these seedlings to these factors really depended on how big you were. So if you were a larger seedling to begin with, you did better overall. And from the traits that we measured, wood density was quite a good predictor for growth. So finally, just some applications from these results. Again, given this kind of size-dependent growth that we saw, planting larger seedlings would be preferable. This is obviously easier said than done, and would definitely increase your initial cost at the start of any restoration project. But if you did manage to get good growth quickly, potentially you might save money later on follow-up plantings or maybe even weed management later on. We did see also an effect of the advantage of being able to grow quickly and establish early. So obviously continually improving any management strategies that really target that area, that early stage of seedling establishment will be beneficial. Finally, while we only use a very, very small subset of plant functional traits to predict seedling growth, I think assessing a large variety of both above and below ground functional traits would be definitely beneficial in the long run, and I suppose with the ultimate goal that we'd be able to tailor species selection to specific site conditions. And with that, I'd just like to thank everyone involved in this project, the landowners, Noel and Penny, collaborators, and especially all the planters and volunteers who spent countless hours running up and down the hills planting trees and measuring seedlings. Thank you.